And now welcome Ravi Chopra to kick off, I think, our lab and clinical perspectives. There he is. Thank you.
Hi everyone. Thanks, Tim. That was awesome. I love the movie. That was really inspiring. My name's Ravi. I'm one of the postdocs and one of the neurologists here, one of the instructors. I come at this from a very different angle from Tim and others. They talked about those instructions, that DNA going to RNA, going to protein, and it's a very vivid reality. That's how these diseases work. I like to think of the brain a little bit differently. Instead of a house, I like to think of it like a flock of starlings that you watch as you drive down the highway. These are complex systems with extraordinary capacities. If we could measure the capacity of this system accurately, maybe we could catch this disease before it is traditionally thought to start. And so what I'm going to tell you guys a little bit about today is the work that I'm doing both in mice and in people to try to prove this. And those are my disclosures. I have none. So, I think Tim mentioned the sort of match that starts the fire. This is often how we think about ALS. It starts right when someone gets sick. And symptoms accumulate progressively, inexorably over time. You see the match, then you see the fire. And there's nothing you can do about it. What we've learned, particularly over the last, I would say, 10 to 15 years, is that this disease started not just days, not just weeks, not just months, but years before the patient ever set foot in clinic. By the time they show up to clinic, it's not a match anymore, it's a wildfire. And so it would be great if we could do something about this when it was a match and not when things were completely out of control. But unfortunately, a lot of our models for ALS care and disease detection are here. This is where neurologists traditionally get involved. What if we could get involved here when the disease actually starts? To do that would require us to recognize the disease before someone has any apparent symptoms and to know for sure why it took so long for this disease to emerge in the first place. Now in the field we have two broad frameworks for trying to understand why does this take so long? And I'm going to use some analogies here to sort of help make this visual to you guys. One is this idea of redundancy. So maybe the reason that you can lose neurons in the brain and spinal cord and not have any symptoms is that the nervous system carries around a bunch of spare tires. So you think about your car, you get a flat, no problem, throw a spare tire on there, car keeps going. This would imply that there was extra in the brain and that it was just there in case you needed it. If you look at how much damage has accumulated in your typical patient at symptom onset, it would be a lot of extra, like maybe half is extra, but maybe that's worth it to prevent the onset of these diseases even earlier. And there might be an evolutionary advantage to that. And this is a totally reasonable hypothesis. But there's an alternate view, which I'm going to call compensation, which is really thinks about the brain more like a household budget. So in your household budget, every dollar is accounted for. Even if you put it into savings or a retirement account, it has some purpose. But what happens if the budget gets cut? You do dynamic reallocation. You go into damage control mode, you try to minimize the impact on your life. So you cut the categories that are less important, and you keep things like rent and groceries intact. And this is potentially how the nervous system could work. Maybe we give up on the ability to learn calculus when we're 50 and we start to lose neurons, but we maintain the ability to walk and breathe. Unfortunately, what we measure clinically is walking and breathing. So what if we had a way to measure the full richness of what a brain can do? To do this, there's an assumption that's built in that I study, which is that a healthy brain tries to be both flexible and efficient, tries to have its cake and eat it too. We have 80 billion neurons in our brain. Those 80 billions are way more than we need to walk. But the ability to do everything that human beings can do rests inherently on the flexibility that's provided by that enormous computational power. If you look at a lot of the AI models like Claude and things like that, they can barely approximate what a brain does. And our brain is a millionth the size of the server farms that are used, and it runs on 20 watts. That's extraordinary. And it does this by threading this needle between flexibility and performance. When the brain gets sick, though, it has to make a choice. And it chooses to preserve performance at the expense of flexibility. It literally covers its tracks by keeping the things that are important intact, like walking, breathing, and it gives up on something, like calculus. If we could measure the flexibility of the brain in real time in a real person, we might be able to catch this disease the very moment it starts. And so in my work, I approach this from 3 different angles. One is to look at behavior. One is to look at the underlying engine of the brain, which is those waves, those oscillations, those sparks that make brain activity actually do what it does. And then one is using wearable sensors. This is an awesome tool for use in patients who either have ALS or are someday destined to have ALS because of an at-risk mutation. So to do this, I've built a lot of tools like this. This is kind of a smart house for a mouse. What it allows us to do is to look at motor behavior with very, very fine precision, but also complete information. This is a little ladder. It's controlled by capacitive touch sensors. The mouse crosses it whenever it wants. I'm also recording from its brain at the same time. The mice love this. On average, a mouse will do this 500 to 1,000 times a day. What's cool about this is I don't tell the mouse how or when or why to do this. It chooses entirely on its own. I'm not asking it to press a lever. I'm not asking it to cross a balance beam. I'm not asking it to run a rotorod. It turns out when mice live in this smart house, they do all sorts of things. We can figure out what those things are just from that capacitive touch time series, and we can put names on those behaviors with very, very high confidence and precision. When we do that, we get a picture of a mouse's life that looks like this. So these are 3 healthy wild-type mice. They're all litter mates, siblings, grew up with the same mouse mom, in the same mouse house, and then at 60 days of life, I put them in the smart house, and they live the rest of their life on that smart house. I want to point out 2 things to you. One is that they're each unique. This is quite cool. That mouse is obviously different from that mouse. The second is that they are quite stable. This mouse on day 50 and 60 and 100 looks the same, but is totally different from that mouse. That's how we can tell them apart. I hesitate to use the word personality because I'm talking about a mouse, but maybe we'll call this motor preference. This is what SOD1 ALS looks like in mice living in the smart house. So I'm going to point out the same thing. They're all unique, but they are all totally unstable. And the red line marks the appearance of weakness. So that instability of their motor world begins months before they show any weakness, almost as if the brain was trying to do something in the context of injury, make some kind of a change, but to keep the mouse walking across the ladder. You can actually take each of those behaviors and look at how flexible, how dynamic are they every single day. On the bottom are my healthy control mice, the ones that I showed you on the left. Sometimes things get more complex. Sometimes they get less. Sometimes they stay the same. Depends on the mouse. But in ALS, inexorably, continuously, long before the onset of symptoms, things get more and more simple every day. This is a literal visualization of sacrificing flexibility for performance. And that's true whether you look at how they explore, how they sit there, how they jump, and how they run. The timeline of this change matches exactly with the timeline of the increase in neurofilament. What that means is that there is no presymptomatic disease. As soon as you lose a motor neuron, you give up something. But what you give up is flexibility and richness. That's actually most of the disease. By the time people are weak, the disease has been there for months. So I don't have time to talk about the other stuff that I do, but the core point I'm trying to get across here is that we can look at behavior in a completely different way. And we can understand this disease as no longer having a presymptomatic period, but what I'll call a post-symptomatic period, where the nervous system is budgeting to maintain performance at a cost of flexibility. We can understand how this happens using concepts from statistical physics. There's a specific one that I'm interested in called criticality, which I'm happy to talk about offline. And I'm building out a wearable program to see if we can measure the same stuff that we're measuring in the smart house in a human being living their life with no intervention from me. And I appreciate everyone's attention and time. I want to give acknowledgments here and say thanks for your attention.